So good morning. Thank you, Herbie, for that introduction. And let me get my computer live. So it's an honor to be here. It's my first visit to Bulgaria. I've immensely enjoyed it. Spent several days in Sofia and then had a long ride down here in a day. But I got to see the countryside and it was quite nice. Before I begin to talk about my research and sort of introducing why I'm so excited about condensate biology, I want to tell you that next year we're going to host this meeting in Cambridge, Massachusetts. So there will be a meeting on June 15th. It will be somewhere in Cambridge near MIT. It's a really nice place to visit in June. And I think the science will be fun. And if you have any way at all of getting there, please do. You'll have a good time. What I'm going to talk about today is condensate biology, a new word, condensate biology. I think it's going to become a very common word, and I believe it's going to redefine how we understand biological systems, not only in how we describe them, but how they are functional and how they actually drive change in biological systems. And I've been in this field for a long time, and I am a little chagrined that having been trained as a physical chemist, that I was so unaware of this until the work of Cliff Brandywine and Tony Hyman in their landmark paper. Well, let's see. Okay. So as you have seen before, this is a diagram or a schematic of the cell, and it illustrates many of the membranous bodies in the cell that now are known to have to be condensates and to be formed by phase transitions and to have properties that are liquid, meaning dynamic, and they exchange components rapidly with with components in the cellular environment. The first body that was recognized to have these properties by Brandywine and colleagues and Tony Hyman is the germ granule in C. elegans, where they recognize that this germ granule, which controls transposable elements in C. elegans, had the properties of a liquid. It would distort, it would fuse and fission. If you photo-bleached it, the components exchanged very rapidly. And then they recognized that the nucleolus, something we have seen for hundreds of years, is also a condensate. As you remind yourself, all the ribosomal genes are located in the nucleolus. They coalesce there, and that's where the ribosomal subunits are assembled. And then you look through histone bodies, nuclear speckles, which we'll talk about in a few minutes. Paraspeckles should be around the speckle. DNA foci have properties of condensates, and in the cytoplasm, you know stress granules has these properties, P-bodies. We now know in RAS signaling, it's condensate biology, synaptic activity. So it is redefining how we see biological and understand biological systems in really fundamental ways. And I've been struggling with some of that biology over my career in trying to understand how gene regulation occurs. And with the understanding that genes are interrupted by introns and exons are assembled in alternative splice ways, the field focused on how transcription initiation starts and then how the splicing process occurs. But little did we know that this initiation process appears to have fundamental aspects that are dependent upon condensates, and as I will talk about briefly, this splicing process also has those attributes in how the spliceosome is directed to assemblin, but we know much less about the spliceosome process than we do about transcriptional processes. Rick showed you this slide yesterday or the day before, talking about how as a vertebrate develops, you generate cell types, and those cell types are dictated in their transcriptional processes and their cell identity by what is called master transcription factors. In this case, as illustrated here in embryonic stem cells, you see super enhancers, clusters of enhancers that are driving these master transcription factors, and they're all interdigitated with one another, giving you feedback systems that then establish the cell identity of an embryonic stem cell. What's interesting about this is that these transcription factors are quite evolutionarily conserved. There's no issue of finding the equivalent factors in mouse and vertebrates. But what's a little known and little discussed attribute is that these enhancers are actually very difficult to see. They're not conserved. They change fairly rapidly. And that gives a specificity to the fact that the transcription factors have a collective way, in my opinion, of evolutionarily collecting binding sequences in a dynamic fashion, which we'll mention later, and driving the transcription that is absolutely critical for all these cell identities and the developmental processes that generate those tissues. Now, I'm going to talk about enhancers and promoters in a very specific way for a moment, because I want to introduce the issue of what is the role of RNA in the condensate formation process that controls the activity of enhancers and promoters and therefore genes. And we know that now that in promoters are sites and enhancers of divergent transcription. That transcription, as I'll show you in a moment, goes in both directions as those sites are activated. This is driven by transcription factors, the master controllers of cell identity. And could this nascent RNA contribute to condensate interaction? And this is a point of contention between Rick and myself, because we just haven't been able to get to the point where we can address that question. What I mean by divergent transcription is that about a decade ago, John List and my lab recognized that when you look at promoter for a given gene, most genes in vertebrate cells, you find a complex of initiation factors that will drive transcription in the gene direction, but you also find polymerase and a similar complex of factors that will drive transcription in the upstream divergent direction. Here we have a cluster of transcription factors, and here we have, over here or down there, enhancers, and they're interacting with this site, and the same process is occurring at the enhancers as it is at the promoters, driven by transcription factors. And we know that after initiation, the polymerase pauses about 40 or 50 nucleotides downstream. A kinase called, a complex containing a kinase called PTAB phosphorylates these negative factors, and that releases the polymerase for further elongation. But we also know that the same process occurs here. So this is not the distinguishing step between the gene direction and the upstream antisense transcript. Now, we've known for some time that this RNA generated in this direction is quickly degraded, whereas the RNA generated in that direction is the gene-specific RNA, and it has to transverse, kilobases upon kilobases of nucleotides to generate the precursor to actually express the gene. So what's the nature of the signal? That signifies that the direction of the gene is the polymerase to be elongated. And we think that signal is the recognition right after initiation, maybe by the polymerase is paused or shortly thereafter, by U1 SNRNP, which is the first step in splicing, and as you know, recognizes the 5' splice site and therefore initiates the splicing process. But it's also well known and was highlighted recently by Gideon Dreyfus's lab that U1 components, proteins, suppress the activity of the cleavage of nascent RNA by the enzymatic activity at these polyvenylation type signals within introns. So here, U1 can suppress this termination of transcription as a polymerase elongates. And I'll show you one experiment in which we inhibit this U1 recognition. We reduce the degradation of RNA, and we ask the question, if we block the U1 recognition, does this RNA terminate transcription and become destabilized due to the exonuclease? I'll get this right in a moment. And this is the result of that experiment. This is the, and I'll take you through it in some detail. Here is the initiation. This is a meta-analysis of all the genes active in an embryonic stem cell. Here's the gene direction. This is first stable nucleosome downstream from the site of initiation. And what you see here under normal conditions is very little RNA being terminated in this region. If we inhibit the exonuclease by eliminating it, we see stabilization of RNA in this region. So the RNA, there's some RNA being terminated here and degraded rapidly. If we inhibit U1 with the active exo, we see a little RNA. So yes, U1 is suppressing this. But if we combine those two, we see a large amount of RNA being generated in this sense direction. Now, if we look up here in the in-sense direction, we see almost no effect of inhibiting U1, that's a green line. But if we remove the exonuclease, we can then see RNA being generated in this direction. So the major signal between gene and non-gene stable transcription, elongation, and not is this early recognition of U1 to the nascent RNA. So here, transcription initiates, we get a pause, and then you have, in this region, U1 recognizing the 5' splice site. It's usually very conserved. There's almost always a 5' splice site observed here. That suppresses termination, allowing elongation. And up here, there's no conserved 5' splice site. So here, coupling of transcription with splicing signals is an early step in defining what a gene is. Now, I told you that this type of divergent transcription is found both at promoters, such as a diagram here, but as well at enhancers. And here we have the possibility that you have RNA nascent, adjacent to promoters and enhancers being generated at the time a gene is being activated. And we know from the work of Mike Rosen and Carl Parker early on in the studies of condensates, that if you have a mixture of RNA binding proteins, as shown here is an RNA recognition with an intrinsic disorder domain, and RNA, which they mix in vitro, you can form a condensate. That condensate, in part, is due to RNA binding proteins giving valency between different RNAs, and the intrinsic disorder domains now forming valency with one another. But there's also recently been recognized that RNA itself can kiss, and that can form valencies that also contribute to the properties of the condensate. The intrinsic disorder domains have lower affinity, and condensates formed with them alone. If you just take this at high enough concentration, it'll form a condensate of the protein by itself. If you have RNA, the dynamics of that process at this moment, as it is constructed with these sort of arbitrary RNAs, are not as dynamic as it is if it has only the intrinsic disorder domain. So, knowing that RNA and protein can form these types of condensates, then one begins to think about transcriptional processes and regulation as being driven by enhancers, and we'll talk about that in the next few moments, being activated by transcription factors, which probably stay on the DNA in long periods of time, promoters that have transcription factors as well, RNA being nascent, RNA binding proteins, splicing proteins, and this whole thing has the properties of a condensate. And we'll talk about that in a moment, but let's begin to think about what some of the things we know about this process of initiation of transcription. Now, Rick Young's lab, for a number of years ago, produced data of this nature, defining super enhancers. These are clusters of enhancers near genes that are expressed at very high levels in cells. In embryonic stem cells, there's approximately 231 genes that have these types of clusters extending over multiple kbs that is defined as a super enhancer by Rick. If you know Rick Young, you know he likes these words like super and master. He's got this complex of being master of the world. And sometimes he is. And then there's this typical enhancer over here, which has one site as an enhancer driving a gene. And you know here, this OX, SOX, and NANOC I mentioned before, you can chip it and it's bound here, and it's bound here at all these enhancers. Polymerase is bound to these enhancers here, it's there. There's a mediator complex that we've known for some time that is complex with polymerase, and it's bound at these sites as well. Cohesion, which you heard about yesterday, is found by chip at these sites. These are modification acylation activities, deacylation activities, and they're all complex at these enhancer sites as well as at the promoter. And then if you look at high-C sort of looping assays, you clearly find evidence that these sites are looping to each other and as well looping to the promoter. And it suggests that this initiation process here, which drives the gene transcription, and as I've argued, also probably has to have splicing components in it, is actually a very complex set of components, because there is an enormous list of proteins that are found sequestered there. So, and then I wanted to point out again that if we looked in other species, the closely related species, these binding sites, a few of them are conserved, but the majority of them are not. So they're dynamically evolving during evolution. So that all raised the question of, is the super enhancer engaging and driving transcription the product of the formation of a condensate? And as you will learn from others who have followed me today, Abraham and Rick and others, yes, the evidence is quite strong now that a condensate forms in the nucleus at the time that transcription seems to be occurring. Those condensates are formed by these binding sites for transcription factors and a whole host of components like mediator polymerase, and a number of other components. And what is striking, very striking, is that for most genes, this condensate is dynamic. It's not stable. It blinks. 8, 10 seconds, 30 seconds, it blinks. So this has an arrow backwards, and it is dynamic in the activity. And in that dynamic control is likely to be the secret to how genes are regulated in a fundamental sense. So we know, and I won't go through any evidence other than this, early last year, that if you look at genes that are known to have super enhancers associated with them, you tag BRD4, one of the components in that complex involved in recognition of acetylated histones. You see it's a foci. You see the gene by in situ hybridization, and they co-localize. And now, for all these genes and for many genes, there is a condensate that's associated with the gene that's active in the production of RNA. Now, these components that form, some of the components in that condensate, that form the condensate have large intrinsic disorder domains. And if you take those intrinsic disorder domains and link them to GFP and concentrate them, they form condensates by themselves. If you, in this case, this is from BRD4, this is a mediator in an intrinsic disorder domain, about 600 amino acids. It itself will form a condensate if you mix them. You mix these two, they will also form a condensate. You mix the carboxy terminal domain of together, they'll form a condensate together. And if you mix the carboxy terminal domain of RNA polymerase, which you know has these 52 repeats of an amino acid sequence that's integral to its activity, it will also form a condensate with these mediators as well as with BRD4. So we can literally, and have just recently published with Rick Chakravarty and Rick and Krishna, who's in the audience, the fact that you can take a cluster of binding sites for transcription factors, shown, exemplified here, mix them with coactivator, BRD4, mediator, and with the carboxy terminal domain of polymerase, but we did it just with mediator, and they will actually form a condensate together, not surprising. That condensate will facilitate the binding of the transcription factor to these sites. So it's cooperative in the formation of a condensate that facilitates the binding of transcription factors to local sites, and this has dynamic activity. So we can form condensates that have many of the components of a super enhancer or an enhancer without RNA, just with DNA and transcription factors. That gives us a concept that you can see condensate biology facilitating the bindings of proteins to the transcription factors, perhaps engaged in the stabilization of those transcription factors and clusters, and then engaging with the polymerase activity, all of the mechanisms unknown, but likely to be the case. Now, after you get initiation of transcription, which we've talked about to this point, about a condensate being involved between enhancers and promoters, engaging the carboxy terminal domain of polymerase. And as I said at the beginning, this carboxy terminal domain in this complex is not phosphorylated, and it forms part of that condensate activity as we visualize it now. We know upon formation of the pre-initiation complex that CDK7, part of transcription factor 2H basal factor, phosphorylates serine 5 in this terminal repeat, as illustrated here. And then after the pause, which I described earlier, the kinase CDK9 phosphorylates the polymerase on serine 2, and that's a signal that correlates with the polymerase elongating across the gene. Well, what's the relationship between this process and the polymerase leaving this mediator process? And what could this actually be involved in terms of the splicing of RNA after initiation of transcription? So what we know in part is we know quite a bit about all these processes, but at a gross level, not a refined level, is that the splicing process, as illustrated here with 5' exon and then the next exon, this does not have to be the first exon, that U1, as I pointed out before, recognizes sequence here, facilitates the binding of U2. Maybe this is not correct, but facilitates the binding of U2. U2 then recognizes the 3' splice site with other components, all of which have serine arginine repeats. And serine arginine repeat proteins bind to exons in exon splicing enhancers and facilitate the binding of other SNRs. And that gives you the splicing process that actually dictates what protein is made from this gene. There are a large number of serine arginine proteins. In fact, there are about 10 of them that are conserved all the way from man back to Drosophila. They are clearly critical as a family for splicing. They are phosphorylated at the serine in ways that are important in the regulation of splicing. We do not know how that occurs. And the serine arginine proteins themselves form a condensate if you concentrate in a test tube. So here is SRSF2 as an example of the SR protein. You have an RNA recognition motif. Some proteins with very extensive serine arginine repeats do not have RNA recognition motifs, but do have very extensive serine-rich, this is amino acid content of this region, and you see serine-rich and arginine-rich sequences. And it's clear from bioinformatic analysis that exon splicing enhancers that bind these serine arginine proteins are the primary signal for the incorporation of that exon during splicing. RNAs binding to the intronic sequences control the rate of that switch between alternative splicing. But the fundamental code for a gene, exon, is these exon splicing enhancers, which are thought to be recognized by the serine arginine protein. Now, downstream of all this recognition, and I won't spend any time, but I'm so thrilled by this, I just have to show it, is that over the last several years, through the work of Yigan Shi, Toshi Nagata, and Reinhard Lerman, using cryo-EM and a fundamental breakthrough in the field, has been able to visualize about 8 different complexes that are involved in the spliceosome cycle to the tune in the catalytic core of 2 to 3 angstroms and to the tune of 5 or so angstroms at regions around, so they can identify maybe 40 different proteins that are involved in this process. There's 11 helicases pushing this complex around, rearranging. It is a machine that is just begging for somebody to do a good model on how kinetically it works. And looking at how they prepared these complexes for analysis, I suspect that we will be able to do in vitro biochemistry to actually test how proteins interact with this component in its formation. As you see here in the cycle, U1 binds and U2 binds. We've known this for 25, 30 years, 4, 5, and 6 binds, and then you rearrange these things, releasing 4 and 6 as small RNAs, and then you get a catalytic cleavage making a lariat, and then you get the second step, and you release the lariat's degraded, all this is great. Now we can see this in atomic restitution. This is amazing. And it is going to lead to fundamental insights. So, how does this occur? Well, we don't know, but we do know that it involves condensates in a very direct way. And here is a immunofluorescent of this protein, SRSF2. You see it's involved, it's concentrated in the nucleus in speckles. That's what the speckles are. They're photobleached, they're dynamic, they exchange components. We've described them as storage sites for splicing factors. The SNRPs are concentrated there. So we have these speckles in the nucleus of an interface cell. If you look, if one of these speckles are associated with a gene, with a super enhancer, yes, they co-localize as shown here. In fact, if you go through a whole series of SR proteins, you can find that they co-localize in part with super enhancers, active genes. And that's not surprising because these SR proteins are the components that are recognizing the nascent RNA and generating the spliceosome that executes the removal of the intron. Now, only about 15% of the SR speckles are co-localized with polymerase. So as I said, these are thought to be splicing sites of enriched splicing factors. And from the work of Rick and Ibrahim, what you see here is the number of polymerases and SR proteins when they do co-localize. And there are approximately 50 to more polymerases in those complexes, and there can be hundreds, sometimes thousands, of SR proteins. So here's another condensate forming on an active gene, transcription, bringing splicing components to this whole process that we do not have the foggiest idea how that works. Now, if you inhibit splicing, you actually, this is a drug that inhibits one of the early helicases, and what you see is that the SR proteins co-localize with the gene in the absence of the inhibitor, but does not in the presence of inhibitors. So clearly these SR proteins are influenced by components that are involved in the splicing of the nascent RNA and the condensate process. So now what we visualize is that the initiation of transcription occurs. As I said, U1 SR proteins are involved in engagement. The polymerase is, as I mentioned before, phosphorylated as it leaves the pause site here, and it's probably actively phosphorylated and dephosphorylated through this whole process. These serine proteins, SR proteins, are phosphorylated, dephosphorylated in dynamic ways. As the exons are transcribed, these coalesce, execute the splicing. The polymerase proceeds. There's a condensate here with many splicing components. This whole splicing component activity is 70 different proteins that go through the shuffling of all these components to have activity. Well, is there any evidence that there is transfer of polymerase from a condensate that's involved in initiation of transcription to a condensate that's involved in elongation? And will be published in recently in Nature. Rick and his colleagues have done the following experiment, where they've taken mediator and mixed it with the carboxyterminal domain, and they will form a condensate together. There's a crowding agent here, but you'll see that here with the carboxyterminal domain they co-localize. If you have the addition of the kinase, either CDK7, that phosphorylates this tail at serine 5, or CDK9 at serine 2, unphosphorylated forms a condensate mixed with mediator, phosphorylated does not. So as I mentioned before, the polymerase with its carboxyterminal domain is involved in condensate formation, can be. But if you look at forming condensates with the SR proteins added with the CTD, here with CTD, this is M-cherry labeled SR protein. It forms a condensate. Unphosphorylated polymerase does not join that condensate. But if you phosphorylate the carboxyterminal domain, they do form a condensate together. Consistent with the idea on phosphorylation that one of the signals for engagement of SR proteins and the whole splicing system downstream is formation of another condensate process that involves serine arginine repeats and facilitates the spliceosome assembly and execution of the process. So what this has meant to me is that it gives us a whole new picture of how these cellular processes occur, how they evolve, how they're regulated, and we know almost nothing about them in detail. And this is a physicist's playground. This is a biochemist's nightmare. And I think it's one of the reasons, and the major reason I'm so excited about this whole process of the engagement of physicists and biologists together in this new way of looking at very important cellular processes. So the work in my lab that I talked about in terms of SRU1 was done by Hiroshi and Anthony, and has been an absolute pleasure over the last several years. We've had a chance to collaborate with Rick Young and colleagues, and this is the people who have been involved from Rick's lab, and with Arup and Chakrabarty and Krishna and Ibrahim and his colleagues, and it's been an absolute delight. And I want to thank you for your attention.
Questions, please?
Yes.
That was beautiful. Since you brought up all
Can
those,
you, because we don't have yet the microphone.
since you had this great picture where you had all the different condensates, can you give me a little bit of insight into the difference of the speckles and the Cajal bodies with regard to the spliceosome?
Well, I'll give you dogma in the field.
Okay, first step.
First step. The Cajal bodies appear to be the sites in which the SNRPs are assembled. And as you see at the end of the spliceosome cycle, 4 and 6 are separated. 4, 6 in the speckles are assembled. And the tri-SNRP is assembled. So it is thought, and there's pretty good evidence for this, that the Cajal body is the site in which that reassembling of SNRPs is involved. So it's a different condensate than the spectral condensates. The spectral condensates are sites where we have historically thought was a storage site for SNRPs and these SR proteins, and that they would be, and they are found adjacent to, as I illustrated here, genes that are transcriptionally active. But there's a site around that speckle called the perispeckle, and that's where the gene is really being transcribed and spliced more than in the midst of the speckle itself. So it's a perispeckle to speckle, and active genes seem to be clustered there. Are all active genes? There's not, but that's where we think they are. And in those speckles, there are two long non-coding RNAs, MALAT1 and NEAT1, that clearly have a role in the whole process that is been studied. There's some data on it, but details not clear.
The questions? Yes?
I think. How much do you envision that during this phase separation, there is actually a conformational change within the proteins in the intrinsically disordered domains that are triggering actually the cluster?
So the question is, how do I envision that during phase transition, these intrinsic disorder domains are undergoing conformational changes? If I knew that, I would be much more famous than I am. So, the affinities there are thought to be, in some cases, from Tony Hyman's work, actually even as low as millimole. These are very transient interactions. In the dynamic condensates, very little is known about them in terms of chemical structure and the nature of them. Steve McKnight has argued that transient beta sheet type formation, very transient low affinity, could be part of the specificity. I mentioned RNA can be part of that specificity. It really isn't known. It seems like something that structural biologists should be able to tell us, but they tell us that looking at something in the middle of a very large complex that doesn't rotate is difficult, but clearly not a lot known. And the specificity of condensate formation is now going to be a major issue, because they all can't be the same. The system does partition by insulator domains and other things, but there has to be specificity in those, some specificity in that condensate formation.
Any other questions? Yes, Peter?
Well, this may be kind of a
Yes, yeah.
philosophical kind of point, but I think it might be relevant to other talks. There's in this genre, there's a long-standing history in chemistry or the physical sciences, actually, between substances with definite compositions and substances with indefinite compositions. It's basically the difference between chemistry attitudes and metallurgy attitudes. And it seems to me that in a sense, the condensate notion is really saying, oh, there's many things that are much more like what we have in metallurgy, where you have lots of different compositions and things like that. So I think the one thing that I'm going to be looking for in the other talks, maybe you guys will tell me, is how much do we know about the compositional fluctuations of these bodies? Do we directly know that they really are not some specific objects that you just haven't really carefully characterized yet, which would be, say, the attitude of a typical crystallographer, that you're just not yet there in getting the complete structure of these objects?
I'll let Rick and Ibrahim answer that question. But I'm absolutely sure we're over here with metallurgy.
Okay.
And we're not as deeply confined by chemistry as we were before.
Any other questions?
Yes. I have a question. So
Just in the pit shop.
you have to bring a lot of these things together, like in the spliceosome condensates or the transcription condensates. That implies there's maybe a limit to how many you can form at any one time. Like if you have only a limited number of these components and they're being concentrated in one place, you can't form, an infinite number of the condensates. Does that limit, or how does that limit the total, rate of transcription or splicing that the cell can carry out? You know anything about that?
The condensate itself, as it forms and can expand and get smaller, is in equilibrium with the dynamic exchange with components around it. So it produces a critical concentration that's absolutely flat, or it's supposed to be. And then at some other site, if condensate forms, there could be a fluctuation. I don't think that's what's there. I think some signal comes to that site and nucleates valency that forms a condensate. So it's at a very constant concentration. But what you point out is true. It is very important that what this means is that within the nucleus of a cell, there is a dynamic exchange of components that are possibly could play roles at a distance in influencing how a gene can be expressed. And I think we may be seeing that with mutations, let's say, that affect RNA splicing and therefore accumulate RNA in the nucleus. And that could be shifting balances of processes by these depletion of or formation of condensates and affecting things at a distance. So I think the nucleus is much more dynamic in that type of activity than we thought before. You can argue condensate buffer it, or condensates are part of the explanation. And in the Hyman work in C. elegans, if you go back and read that paper, there is clearly regulation in C. elegans in these germ granules about the movement of components between condensates.
Jose? It's very, very nice to see all these things, but I have a question just following on Peter's point of view, just to continue the discussion. If these condensates are used in terms of bringing all these components together, but we still the metallurgy aspect, is that sufficient for all the regulation you need to have a precise gene expression, or something else has to take place? It's like the condensate is just a way of collecting the piece together, but then there's additional mechanisms that make it very specific, or is that sufficient?
My colleague here,
Aru.
answer.
So these are
Stand up.
These are not equilibrium structures, and so what happens after the condensate is formed in ways that we fully don't understand right now, are there are fluxes that are created that lead to partitioning of molecules into it, transport to particular sites, and regulation happens those ways. So it's not, first it's just concentrating it, and then there are non-equilibrium fluxes that lead to various long-range information transfer, if you like, that allows this to happen. These are not, just, these are actively controlled, driven phase transitions, not equilibrium ones.
Any other questions?
Well, let me add to this.
Let's get the microphone.
Yeah, so please.
We'll get to microphones.
This is one of the reasons I find this whole thing so exciting, because that we don't know what nucleates in a non-equilibrium way and how these condensates are involved in that process to give transcription regulation. But until we understood this process, I don't think we can get to that process. But we're now in a stage, I think we can begin to get to that process. But you're right, this equilibrium process is not the nature of its component in the regulation, that's clear. But it's not the fundamental, in my opinion, event.
That's very nice.
Yeah.
So, Claudia?
So RNA is kind of acting like a glue in all of this. I was just wondering where you envision the DNA polymer itself playing a role in condensation, or if it's always peripheral to like the, I guess, center of mass of the actual condensate. And also, do you think these condensates have any solvation properties that would affect, like solvent-like properties that would affect the structure of the DNA that could impact maybe how these proteins are interacting with the DNA?
Let me just give you
So the interesting thing about all of cell biology is that DNA and the binding sites of factors that bind to DNA is really the key to all these events as to where it happens and how it happens. So that's really a fascinating. DNA beyond that has no role as far as I know. The cell doesn't want DNA to be involved in metabolism or many things because DNA is where it stores its information. So DNA binding to factors that then form a condensate, or we heard yesterday involved in loop formation by CTCF binding, is a component of where these occur. Now the question, you're saying, does the condensate itself, we have to think about these condensates aren't, once they form, it's not a homogeneous cellular environment. There's chemical concentration in that condensate, and anything that binds to that condensate preferentially, or components of the condensate, should be concentrated in that condensate. So now even small metabolites could possibly be envisioned to be concentrated in a condensate. And that could facilitate phosphorylation, modifications, rearrangements, or even dissociation. As Tony Hyman has shown, ATP can be a hydrotrope at high enough concentrations to dissociate a condensate. So it not only engages macro components, it could also engage chemical components involved in metabolic activities in cells.
So last question. By the way, this microphone works.
Oh,
Yeah,
okay.
so if you can answer, yeah, like this. So last question.
Hi. So I'm trying to think of this as from functional point of view, but basically what you're saying, you have the separate condensates along the gene, right? You have the initiation and then elongation. And my question is like, so the transcription pausing site where the RNA polymerase is pausing is the place where RNA pol exits the initiation condensates and then goes to the elongation. Okay, and actually my question is, if you remove the pausing site, then would you have one condensate? So
Not, well, okay. Not likely. Not likely because the polymerase is not known to be able to elongate across the transcription unit without being phosphorylated. So there are promoters in which there's very little pausing. As soon as the polymerase initiates, it appears to be phosphorylated and then move. But then it would, by phosphorylation, begin to be engaged in the engagement with the SR factors. That's the way we visualize it. Whether that is universally true, there's probably not a universally true hypothesis in cells, but that is the general conceptual track. Thank you very much.
So let's thank again Professor Chuck. We could have kept